Um, Welcome everybody. My name is David Ban. I'm joined by my colleagues Gemma and Tim, and today we'll be introducing our genetics resource at the Centre for Longitudinal Studies. I'll be giving a brief introduction to the cohorts. I appreciate that many of you may not have heard about the cohorts before. You might be wondering what is the value to research that you could get from these kinds of studies, incorporating genetically informed designs, in contrast to sort of large-scale studies you might be more familiar with, like UK Biobank. Gemma will be introducing our genetic resources, and Tim will be giving an example of ongoing research in this space. The overarching aim is to give you information on the data that we are making available to the research community free of charge, and to hopefully stimulate some research ideas that you might be developing. A little bit of housekeeping: we are recording this session, so that means it will be available online at a later date. If you have a question, please add it in the chat, and we'll be doing Q&A at the end. For technical issues, please email this email address, and then please also fill in the feedback form at the end. Thank you. Okay, so the Centre for Longitudinal Studies, about our cohorts then. So in a single week of 1958, all the children born were enrolled in a longitudinal study. They gave data when they were babies, and they've been giving data across their infancy, childhood, adolescence, and across adult life into old age now. This is the birth cohort design. So we have one birth cohort started in 1958. We have another one that started in 1970, again with follow-up across life. We have another one that started in the millennium with follow-up across childhood and adolescence and now early adult life. And we have another study that was much like Alsbach, was born around 1990, but this was followed up from adolescence onwards. This is called Next Steps, and this is designed to be representative of England. So this snapshot of the study timelines gives you an overview of the time points at which we have data in each of the different studies. So we actually have data from birth from each of the birth cohorts, and we have data from across childhood, adolescence, and across adult life. So we see that there's a great span of data that is collected, and so this may be stimulating you to thinking about how you can use the longitudinal nature of these studies, incorporating that in with a genetically informed design. For example, you could be looking at how genes correlate or cause outcomes across the entirety of life. You might be thinking about how gene-environment interactions may differ across time. You may also be able to investigate genetically informed designs comparing different cohort studies. We have over a 50-year time span here in which you can compare how, for example, gene-environment correlations or gene-environment interactions may change across time. So lots of data across the time span and across a wide chronological window. We should mention here that the 1946 is included in this graph, and we are hopeful that in the future this will be included as part of the Centre for Longitudinal Studies going forwards. This again is a birth cohort study design with data across life. Each of these studies is designed to be representative, nationally representative of Britain, or of England in the case of Next Steps. And so we also provide guidance for individuals wanting to account for loss to follow-up across time. Like all longitudinal studies, we tend to miss individuals, they tend to drop out of the study as the study goes on. But the good thing about the cohorts is that because we have the early life data, so we can use this information to make, in our principled approaches to missing data, to try and reduce bias due to attrition. And we give guidance and we give syntax on our website for this kind of thing. This is a snapshot of what the kinds of data that we collect here in the NCDS, so the 1958 birth cohort study. We have anthropometric data, height and weight, measured across life. We have a biomedical data collection that took place in midlife, containing blood samples, biomarkers, and blood pressure. We have mental health and well-being and happiness data across life. We have cognitive measures in childhood and in midlife. We have area-based measures measured on a few occasions. And we have consent for health and economic linkages as well. This slide shows the kinds of information that we have on each of the different cohorts. So we can see data from birth, on the birth weight, labor measures, and pregnancy. We have social data, we have health data, and we have data on continued qualifications and skills. I think more broadly that the message here is that we have very rich health and social data and psychological data across life in many different cohort studies. We have three data collections that took place in succession during the COVID pandemic. We used a harmonized questionnaire for each of the different cohorts that we run. We also have serology data collected as part of this. Please see our website for more information. These data, like all of our data shown previously, is available for free on the UK Data Service. We have increasing amounts of linked administrative data, both for health and education. So this slide gives a snapshot of this. I think it's better for you to read separately because there's quite a lot of information. But these data are available on the UK Data Service by using their Secure Lab feature. And in terms of using genetics with this data, the pathway would be importing derived genetic data into the UKDS Secure Lab. For example, importing in polygenic scores that you may have derived. Our website contains an awful lot of useful information. We have a training and support program of work. So we have pre-recorded webinars giving guidance on each specific study. We have overviews of specific types of data, like well-being data, or data for aging, and our data on care measures. And we have a webinar on giving guidance for missing data as well. Upcoming training events include one on conducting cross-cohort comparative analysis that will take place in May of this year, and another on handling missing data using BCS 70 that will take place in June of this year. Please see our website for more details. Tim, who is going to be presenting later, will be leading a webinar on using polygenic scores in the cohorts later on in this year. So these data, as you'll hear in a moment, will be made freely available to all bona fide researchers via the UK Data Service.
Can everyone hear me? Can you? No, David, if you nod.
I can hear, yeah, thanks.
Yeah. Okay, great. So I'm just going to tell you a bit about the genetics data we have here at CLS. So here I've highlighted in green the cohorts which we have genetic data for currently. So we have genetic data which we are sharing for NCDS, BCS 70, and MCS. And the reason next steps is in orange is we have collected samples for next steps, but they haven't yet been genotyped. So we're hoping for that to happen at some point this year, and then maybe we'll make the data available by the end of the year. So here is a little overview on the data availability we have for each of the data sets. So for MCS, we have a genotype and whole exome sequencing data. We have genotype, imputed data, exome data, and epigenetic data. For BCS 70, we have genotype, imputed data, and epigenetic data. And as I just mentioned, for next steps, this is currently in prep. So we don't have any data that we're currently sharing, but hopefully will by the end of the year. As well as that, we're currently generating polygenic risk scores across our cohorts, and we're also deriving epigenetic clock measures. So the majority of the genetic data we have has been generated using microarray technology. So on these arrays, hundreds of thousands to millions of genetic variants for a single individual can be assayed at one time. And it's worth noting that although they don't contain all of the genetic variants in the DNA sequence, they do have genome-wide content, and they generally include sites which have clinical significance. So I'll tell you a little bit about the MCS genetics data. So these samples were genotyped at age 14. So this was the cohort members. But in addition to the cohort members giving their samples, their parents were also asked to give samples. So we have data for over 21,000 individuals. And these data were generated using the GSA array. So there's over 600,000 genetic variants for this data. And we also have imputed data. So in genetics, this is the statistical inference of unobserved genotypes. And the reason we do this is to boost power and coverage across the genome. So the reference panel we've used is the TopMed reference panel. So this is the most recent and largest reference panel. and it's improved the identification of rare and low frequency variants. And it also works better across a range of ethnicities in comparison to previous reference panels. And this is really great for NCS because they have oversampled underrepresented people. So there's like a lot of range of ethnicities within this data set, although to represent the UK. So this is a breakdown of the imputed and QC'd top med data. So we can see there's around nearly 100,000 cohort members, a similar number of mothers and slightly less fathers. And the number of complete trios is just over 3,000. So as well as the genetic data for MCS, we also have whole exome sequencing data. So the exome reflects the protein coding portion of the genome. And although this represents less than 2% of the genome, it contains over 85% of known disease-related variants. So this makes it a very cost-effective way of generating data, like in comparison to whole genome sequencing data. And it also has an advantage over just the genotyping arrays in that it has much higher sensitivity to detect low-frequency variants. So Sanger did the generation of the whole exome data for us. So this was for over 15,000 samples. And after QC, we have around 14,700 samples and nearly 2 million sites. So again, this is the breakdown of the child, mothers, and fathers in this data set. So there are some great advantages of using the MCS data. So because it's a trio design, you can look for parent of origin effects. So you can see if things are coming from the mother, so maternal effects, or the father, paternal effects. You can also identify de novo mutations. So this may be a mutation which arises in the child, which isn't present in either parent. And as well as that, you can look for gene-environment interactions. And something else a lot of people are interested in are indirect genetic effects. So this could be the genetics of the parents, which influence the child without the child actually having the genetic variant themselves. So now I'll tell you a little bit about the NCDS genetic data. So we also have genotype data for this. It's slightly more complicated than MCS in that the data was generated over many, many years across 7 different genotyping arrays. So a lot of these are kind of older arrays, which aren't necessarily used so much these days. But there's around 6,400 unique individuals across all of these different arrays. So what we focused on doing is combining the data for these into one data set, which is a lot easier to use. Because another issue with the data was they're all kind of on older genome builds. So we made sure they're all onto the latest genome build. So we've imputed the data and quality controlled it and imputed it using the top med panel. And after QC, we have around 6,380 individuals and around 7.5 million genetic variants. So there's slightly less genetic variants than the MCS data, but this is kind of what you would expect when you're combining across so many different genotyping chips. So as well as that, we have some exome data for 1,000 individuals, and we also have some epigenetic data. So epigenetics are essentially chemical markers which go on top of your DNA, and this controls how genes are regulated. So the epigenetic mark which we have is DNA methylation. So this is the most common epigenetic mark. And essentially, when a gene is methylated, the gene is switched off, there's no transcription. And when it's not methylated, the gene is switched on. So we have 2 batches of DNA methylation data in NCDS. We have the batch 1, which has around 540 samples. And we have batch 2, which has around 1,400 samples. And what's interesting about this is we also have 2 time points for this at ages 45 and 62. And the thing about epigenetics is, in comparison to genes, which obviously can't change, you're born with them, it's actually dynamic. So there could be changes within these 2 time points. So this data was generated using the Illumina EPIC array. And after quality control, we have around 800,000 DNA methylation sites. So here's a bit of an overview for the BCS70 data. So we have genotype data for over 5,800 individuals. And again, this was genotyped using the GSA array. And there's over 650,000 genetic variants. So this data has also been imputed, QC'd, and it used the top med panel. So one of the focuses we had was trying to get all of the genetic data QC'd using the same pipeline and also imputed using the same reference panel. And this is kind of to create harmony across the data set and make them easier to use together to make them more comparable. So after quality control, we had around 5,600 individuals and around 8.6 million genetic variants. So we also have some DNA methylation data for BCS70 for 255 samples. And again, there's around 800,000 DNA methylation sites after quality control. So we're currently generating polygenic risk scores for our data sets for each of the cohorts. So what's been discovered is that many traits are polygenic. So this means they're caused by many genes or small effect. So PRS essentially summarize into a single score the number of genetic variants an individual has which are linked to a particular trait. So if you have less risk genes, you'll be on the left side of this distribution. And if you have a lot of risk genes, you'll be on the right end of the distribution. So as you can see, it kind of falls into this normal shape, and the majority of people kind of fit into the middle of this. So there are differences between case control groups and PRS, and this has been shown over and over again, although as you can see, the distributions overlap. So we're generating PRS in-house using a couple of different methods. And as well as that, we're using the polygenic index repository. And we're going to be generating PRS for a range of different health and social traits. So as well as that, we are deriving epigenetic clock measures for our data. So essentially, epigenetic clocks use DNA methylation data to create biomarkers of aging. And if you have positive epigenetic age, you're essentially biologically older than you should be. And if you have negative epigenetic age, you're biologically younger than you would be expected to be. So there are actually two generations of epigenetic clocks. So there's the first generation, and these largely correlate with chronological age. And this includes the Horvath and Hannum clocks. And then there's also the second generation epigenetic clocks. And these incorporate additional biomarkers of aging and are designed more to predict lifespan and health span. And these better reflect biological age and age-related diseases. And this includes clocks such as pheno-age, Grimm-age, and the Dunedin-Pace clock. So we're going to be generating epigenetic clocks for all of the DNA methylation data we have. So this will be for NCDS and BCS70. And we'll be generating epigenetic clock measures for all 5 of the clocks that I just mentioned. So the first generation clocks, the Horvath and Hannum ones, as well as the second generation ones, the Grimm-age, pheno-age, and Dunedin-Pace. Now I'll just provide a little bit of information about how you can actually access our data. So the link at the top is where you can download the form, and it will tell you a bit more about how to fill it out. So you can fill out the form and you apply to CLS directly, and then your application will be assessed by the monthly data access committee. And we currently aim to get our data out within 3 months of the application. We also have a lot more information on the quality control and the availability of the CLS genetic data on our CLS GitHub page. And as well as that, as David mentioned earlier, we will be running another webinar on the polygenic risk scores once the repository is finalized. So you'll get more information about that in the year if you're interested in attending that too. So this is just a snapshot of the application. It's just to let you know that when you fill out the form, you can actually apply for all of the cohorts in one application. They're just tick boxes. So that's a useful thing to know. And in the future, we're hoping to have the PRS and the epigenetic clocks made available via special license on the UK Data Service, which David was talking about earlier. So the PRS should be available on the UKDS by the end of the year, and the epigenetic clocks should be available next year. And if you want to apply for these before these dates or not on the UKDS, you'll have to go through a DAC application. So that was the form that I mentioned on the previous page. So this is just a little snapshot of our CLS GitHub page. So this is where it contains more information about the genetics data and the quality control. So it's got a bit of information about each of the cohorts and the polygenic risk scores and epigenetic clocks. I am now going to hand over to Tim, who's going to tell us a little bit about what kind of research you can do using these studies that we have.
Yes, as Jem said, I'm just going to touch very briefly on some of the kinds of analyses that could be done with the genetic data that we've got here at the CLS. So I'm going to use the example of trios within the Millennium Cohort study, which has just come up a couple of times in the question. Sorry, in the Q&A. Advantage here, I think, is that MCS is the only UK nationwide study which has genotyped trios. It was very carefully designed, so it's representative at baseline, and there's a lot of work that's been done that's all available on the CLS website, which has been led by Richard Silverwood, on using weighting to kind of ensure that the sample representativeness is kept throughout the subsequent waves. So there's kind of potentially a really powerful tool here. And so the potential example I'm going to talk about is environmentally mediated genetic effects. And so we can consider that children, or we all inherit both our genes and our environments from our parents, but these kind of genetic and environmental effects on traits and phenotypes of interest aren't necessarily kind of unique, separate, and purely additive. They can differ in quite complex ways. So there might be gene-environment interaction, as David mentioned. There could be moderation of social environment. There could also be this kind of contamination, if you like, of genetic and social effects. So we might have social effects that appear in the genome. So we think they're kind of true genetic effects. We see this with associative mating and indirect genetic effects. And there might also be genetic effects kind of masquerading as social effects if we see differential kind of genetic risk by different groups or cultures, and what we tend to see with population stratification bias. And so to get around this, we need family data. So we can think about potential questions we might want to ask, such as how does one's genotype affect their BMI throughout life, or how does their genotype affect some aspect of their education? And let me just turn on my laser pointer. I hope you can see that. So for any individual, we can consider that their genotype putatively impacts their phenotype, but reality is a bit more complex. And we can think of any individual's genotype as being the combination of the transmitted maternal genotype and the transmitted paternal phenotype. So my genome is half of my mum's genome and half of my dad's genome. Now we can use this, but there's kind of also there's various ways we can do this, and there's some potential nuisance parameters. So if we think about kind of drawing out a fuller DAG, we can see that the transmitted effects, so the parts of my genome that I receive from my parents, can also operate through the social environment, so the family environment, through phenotypes of my mother and my father. And there can also be, as Gemma touched upon, these non-transmitted effects, whereby the bits of the genome that I don't receive from my parents can have an impact on the environment that I receive, that I grow up in, which can in turn impact upon my my outcome, my phenotype. And we can look at mediation of these direct genetic effects through these pathways, and we can also look at, in theory, these non-transmitted genotypic effects through the environment. By definition, they're not transmitted. They can't, the bits of my parents' genomes that I receive, they cannot have any plausible biological pathway through me because I don't receive them. So they can only operate through the environment. And so we could ask, for instance, with respect to BMI, how does the food environment that children are reared in affect their later life BMI? We could look at differential maternal and paternal effects to get at potential intrauterine effects, which can, or should in theory, or rather can only operate through the mother rather than the father. We might apply these to education to look at aspects of the household learning environment or parental investments in kind of child rearing environments. The classic, albeit horrible and a basic example here, for instance, is books in the household and kind of the nurturing educational environment that parents might give to children. And what we can do is we can stitch together, so the transmitted genotypes already exist because they're the offspring in the MCS, so the main cohort member genotypes, but we can also stitch together the maternal non-transmitted genotypes and the paternal non-transmitted genotypes to create pseudo offspring, which can be used for analysis of these indirect effects. And that kind of allows us to explore pathways of mediation in a way that we can't do with data sets that don't contain family data. I can't see what my next slide is. Okay, here we are. So in the MCS, as Gemma touched upon, there's just over 3,000 complete trios. There's, off the top of my head, I think it's another 2,000-3,000 offspring mother and offspring father duos. So we can potentially use imputation of parental genotypes to kind of bulk up that number. And the education example gives, I think, really nicely shows the value of MCS, because we're able to use data linkage to the Department for Education's National Pupil Database. That's, for those of you who don't know, that's the kind of administrative record of education for every child, or almost every child in England and Wales, I think, as well, which contains information on their schooling, their school environment throughout childhood, and also on their the really quite precise exam scores at every major key stage of education, so from kind of age 7 to age 16. And we can use these data through the UKDS Secure Lab, which is a trusted research environment. We can pull in some of the really rich phenotypic data measured across the many waves of the MCS. So this might include age, sex, parenting behaviours, socioeconomic background. And we can also pull in polygenic scores that we've derived from the main genotype data. And for anyone who wants this, we can touch a little bit about the Secure Lab process, but essentially you are able, or we at CLS are able, to derive polygenic scores of interest that can then be imported into the trusted research environment of the UKDS. We've got some of this work ongoing. We're not going to discuss in detail, but kind of briefly on the BMI side, we see that there are maternal indirect effects and not paternal indirect effects. So again, it's supporting that an intrauterine effect net of own genetics. And in the educational context, we see the presence of these indirect genetic effects, which are around a third to a half the size of the transmitted genetic effects, which again, pretty consistent with previous work. And we're able to look at the pathways, or some of the pathways through which these genetic effects, intergenerational genetic effects, arise. So that's the end of my bit. What first to say, please follow the link to the feedback survey. It's really important to us. It's this is the first time we've run this, and your feedback allows us to kind of build on the webinars and improve the content that we're providing. So please do that. We'd really appreciate that. And now we've got half an hour-ish for any questions that people have.